Now, some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other. Example: linear strings or spheres. Spheres arranged upon each other in the form of cube or plate. So, is the architecture of a protein otherwise called the quaternary structure of a protein? Adult human hemoglobin consists of four subunits. Two of these subunits are identical to each other. Hence, two subunits of alpha two subunits of alpha type and two subunits of beta type. Two of these are identical to each other, right? Hence, two subunits of alpha type and two subunits of beta type together constitute the hemoglobin, right? Hb. Now, the last thing is the enzymes we are going to study. Almost all enzymes are proteins, right? These are there are some nucleic acids that behave like enzymes. These are ribozymes, right? One can depict an enzyme by a diagram line. Now, like an protein, they also have a primary structure like amino acid sequence of the proteins. Then an enzyme like any has the secondary and tertiary structures. So when you look at the tertiary structures upon itself, the chain crisscrosses itself. The chain crisscrosses itself, and hence many services or pockets are made. Okay, one such pocket is the active site. Now, active site of an enzyme is a service uh, or pocket into which the substrate fits, right? Thus, the enzymes through their active sites catalyze reactions at a high rate. So, these are the active sites that catalyze the enzymes at the high rate, right? But one of the major difference between inorganic catalyst and enzymes, because inorganic catalysts work efficiently at higher temperatures and higher pressures, while damaged enzymes get damaged at higher temperatures, say above 400 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperatures, that is, hot winds and sulfur springs, are stable, and they retain their catalytic power even at high temperatures upon 80 to 90 degrees Celsius. Thermostability is thus an important quality of an enzymes isolated from thermophilic organisms. Now the chemical reaction. So two types of changes occurs in chemical compounds: a physical change, and then there is in physical change, change in shape without breaking of bonds. This is physical process. Change of state of matter, another: ice melts into water, okay, when water becomes vapor. And when the chemical bonds are broken and new bonds are formed during transformation, okay, this is called as chemical reaction, right? For example, BOH2 plus H2SO4, BSO4 plus 2H2O, right? So this is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. So rate of a physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate del P by del T. Okay, rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature among other factors, right? A general rule of thumb is that a rate doubles or decreases by half for every 10 degree Celsius change in either direction. Okay, catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. So CO2 plus H2O, that is carbon dioxide plus H2O, in presence of carbonic anhydrase, they form the carbonic acid, that is H2CO3. Okay. So in the absence of any enzyme, this is very slow reaction, but like 200 molecules of H2O being formed in an hour. but when the enzyme is used, like 6 lakh molecules being formed in every second, that increases the rate by almost 10 million times. When each of the steps is catalyzed by the same enzyme complex or different enzymes is called a metabolic pathway. Okay, there are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. Multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzyme, is called a metabolic pathway. For example, glucose converts into 2-pyruvic acid, C6H12O6 plus O2 gives C3H4O3. Okay, C3H4O3 plus 2H2O. Okay, so is a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes. Becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. Now in our skeletal muscle, under anaerobic conditions the lactic acid is formed. Under normal aerobic conditions pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, that is alcohol. So hence in different conditions different products are possible. Like now another context is how do enzymes bring about such high rates of chemical conversion? Because they have the active side, right? The chemical or metabolic conversion refers to a reaction. Now chemical which is converted into a product is called a substrate. Hence enzymes, that is proteins with three-dimensional structures including an active site, they convert a substrate into a product. Okay, S to P, right? Now S is a substrate, has to bind to the enzyme at its active site within a given cleft or pocket. Now substrate has to diffuse towards the active site. There is thus an obligatory formation of enzyme substrate complex. Obligatory formation of an enzyme substrate complex is formed. Now this complex is transient phenomena, temporary phenomena, right? During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Okay, so transition state structure is formed. Now very soon after the expected bond breaking or making is completed, the product is released from the active site. Okay, in other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state structure. Okay, so there could be many more altered structural states between the stable substrate and the product. So there could be many altered structural states. That all the other intermediate structural states are the unstable, right? Stability is something related to energy status of the molecule or the structure. Hence we look at this, we see the potential energy, right, for the stability. So potential energy and progress of reaction, right, is structured, and then there is a product. So there is a so-called state, that is transition state, and between the substrate and product there are many more altered states. All these are unstable, right? So activation energy without enzyme, it has a larger barrier and smaller barrier activation energy with enzyme. So this is all the diagram. Now y-axis represents the potential energy and x-axis represents the progression of the structural transformation or states through which the transition state. Okay, Now the energy level difference between the substrate and product. If P is at the lower level, then yes, means product at the lower level, then S, the reaction is an exothermic, that is energy is being released. Now okay, however, whether it is an exothermic or spontaneous reaction or an endothermic or energy recurring reaction, the substrate, that is S, has to go through a much higher energy state or transition state. Okay, the difference in average energy content of substrate from that of this transition state is called the activation energy. Now the average difference, when you take the average difference of the energy content between the substrate and this higher energy state, that is transition state energy state, that is a transition state, higher energy state, that is a transition state, is called the activation energy. Now enzymes eventually bring down this energy barrier, making the transition of substrate to product is with more ease, right? Now nature of enzyme action. Each enzyme has a substrate binding site in its molecule so that a highly reactive enzyme substrate complex is produced. So each enzyme has a substrate binding site in its molecule so that a highly reactive enzyme substrate molecule complex is produced. So enzyme substrate complex is a highly reactive, right? It is unstable. Now this complex is short-lived and dissociates into its product P, and the unchanged enzyme with an intermediate formation of enzyme product complex, and the unchanged enzyme, so it dissociates into product and unchanged enzyme with an intermediate formation of the enzyme product complex, that is EP. So first enzyme plus substrate, they get closer, they form the enzyme substrate complex. It is short-lived, it is transition state. Then there is, so before it gets converted into product, and then when the product is released and unchanged enzyme is released, before there is another stage, that is EP, that is enzyme product formation. Okay, so this is what it's written in here. The catalytic cycle of an enzyme action can be described in the following steps. Now this is important. First, the substrate binds to the active site of the enzyme, fitting into the active site, right? First, fitting it on the active site. Now second, the binding of the substrate induces, now this binding, when the substrate binds to an enzyme, now this binding induces the enzyme, right? It induces the enzyme to alter its shape. Now the enzyme altering its shapes so that fitting more tightly around the substrate, so that the enzyme gets fit more tightly to the substrate. So substrate induces all this, but enzymes show more tightness, right? After that, after the after the induction by the substrate, the active site of an enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product form is formed. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and make a new bond, and new enzyme product complex is formed. Now the fourth step, the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Okay. Now the factors affecting enzyme activity, right? The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. This include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulates its activity. Okay, I included factors that affecting enzyme activity. These are temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Now temperature and pH. Enzymes generally function in a narrow range of temperature and pH, right? Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and pH. Optimum pH, right? Now activity declines both below and above the optimum level. Like low temperature preserves the enzymes in a temporarily inactive state, whereas 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 high temperature destroys enzymatic activity. whereas high temperature destroys enzymatic activity because proteins are denatured by heat. So this is it, like temperature and pH. Now talking about the concentration of substrate, like enzymes are specific in nature, so change in the substrate concentration, it affects the rate of reaction. Now concentration of substrate, like with the increase in substrate concentration, the velocity of the enzymatic reaction rises at first, right? The reaction ultimately reaches a maximum velocity, that Vmax, which is not exceeded by any further change in concentration of the substrate, right? So it gets constant. But this is because, and why is this happen? This is because the enzyme molecules are fewer than the substrate molecules. So even if you change the concentration of substrate, since the enzymes molecules are fewer, not available, so, and after saturation of these molecules, and when these few molecules get saturated with the kind of substrate molecules, after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. Okay, the activity of an enzyme is also sensitive to the presence of a specific chemicals that bind to the enzyme. Now the activity of an enzyme is also sensitive to the presence of the specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition, and the chemical is called the inhibitor. So chemical inhibitor and process is called inhibition when shuts off the enzyme activity. Okay, now okay now now when an inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Now due to its close structural similarity with the substrate, now due to this close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are also open in the control of bacterial pathogens. Now, as I told you, like consequently the substrate cannot bind, and as a result, the enzyme action declines. Example: inhibition of succinic dehydrogenase. Inhibition of succinic dehydrogenase, it is an enzyme, and the inhibitor is malonate, which closely resembles the substrate succinate, and substrate is the succinate in the structure. Okay, so this is an example of competitive inhibitor when the substrate molecule closely resembles the structures. Okay, now the classification and nomenclature of enzymes. Okay, there are enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named accordingly by the 4-digit number, right? Now oxidoreductase dehydrogenases, the first class. Okay, the first class, right? Oxidoreductases and dehydrogenases. Now what does this class do? Enzymes which catalyze oxidoreduction between two substrates S and S dash. First, S is reduced. After the reaction, S is oxidized. S dash is oxidized. After the reaction, S is reduced. So it's opposite, right? So this is oxidoreductase and dehydrogenases. Remember, it is dehydrogenases, oxidation and reduction. Now, second class is transferases. Now, enzymes catalyzing a transfer of a group, that is G, other than hydrogen, between a pair of substrate, that is S and S dash. You see, S is attached to G, but after the reaction, S is separated and S dash is attached to G, so it is transferase. There is transfer of a group, but hydrogen is not counted as a transfer of group. Okay. Now the third class is the hydrolysis. Now enzymes catalyzing hydrolysis, that is breaking, attack of water and breaking the compound, right? Now hydrolysis of ester, ether, peptide, glycosidic C halide, PN bonds. That is PN, that is double bond. So attack of water and it breaks it. Okay, so enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic C halide and PN bonds. Now lysis, that is fourth class. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Now see, enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Okay, so X and Y gets converted, leaving the double bond, so it is a lysis but other than hydrolysis. Okay, other than hydrolysis, right? Other than hydrolysis, right? So this is the lysis, and then fifth class is the isomerases, includes all enzymes catalyzing interconversion of optical, geometrical and positional isomers. Okay, it's kind of chemistry. Isomerases. Okay, now sixth is the ligases. Now enzymes catalyzing the linking together of two compounds. Okay, linking together of two compounds. Example, enzymes which catalyze joining of CO, CS, CN and PO bonds. CO, CS, CN and PO bonds. 4 examples are given of ligases. So I said like classification and nomenclature of enzymes. You study like 6 classes. First is the oxidoreductase and dehydrogenases. Second is the transferases. Third is the hydrolysis. Fourth is the lysis, and fifth is the isomerases, and sixth is the ligases. Now talking about the cofactor. Enzymes are composed of one or several polypeptide chains. So they are composed of one or several polypeptide chains. However, there are number of cases in which non-protein constituents called cofactors. Now cofactors, these are the non-proteinaceous constituents, are bound to the enzyme. Now these are bound to the enzyme to make the enzyme catalytically active. Okay, catalytically active. In these instances, the protein portion of the enzyme is called. Now enzyme has the protein part and some non-protein part. Now the protein part is called the apoenzyme. Now three kinds of cofactors, now which is a non-protein part. So three kinds of cofactor, that is, first is the prosthetic group, second is the coenzymes, and third one is the metal ions, right? Now prosthetic groups, these are the organic compounds. Coenzymes also organic compounds, but what's the difference between two of them? Are yeah, the prosthetic groups are distinguished. The prosthetic groups are distinguished from the other cofactors, that is coenzymes, in that they are tightly bound to the apoenzyme. They are tightly bound to the protein part. Now, for example, in peroxidase and catalase, these are the enzymes which catalyze the breakdown of hydrogen peroxide to water and oxygen. Now heme is a prosthetic group and it is a part of the active side of the enzyme, right? Now coenzymes are also organic compounds, but their association with the apoenzyme is only transition, right? Coenzymes are organic compounds, but their association with the apoenzyme but their association with the apoenzyme is only transition. It is not tightly bound or something. It is only transition, usually occurring during the course of the catalysis, right? Furthermore, coenzymes